Welcome and thank you for joining me for this GJE expert talk. My name is Ross Cummings and I'm Director and UK and European Patent Attorney at GJE. I work with biotech, pharmaceutical and medical device companies, assisting them to devise and implement effective patent strategies to support investment in their new technology. So far in our expert talk series, you have gone through the fundamentals of patents, designs, trademarks and unregistered IP rights. And most recently, you've been introduced to the fundamentals of patent portfolio strategy and lifecycle management, where you heard about how to put policies in place to facilitate the identification and capturing of new IP. In this talk, I'll be picking up on some of these themes as they relate to the fields of chemistry and life sciences, in particular biotechnology and pharmaceuticals, which is my main area of practice. Now, this talk is divided into two sections. In this part, I'll be talking about the first pillar in knowing when you are ready to file your patent application, and that is knowing what can be patented. So I'll be discussing the types of invention that can be patented in biotech and pharma, along with what is prohibited. And my focus will be on European practice, but I will mention the US when the practice is significantly divergent. So what can be patented? The patents are a property right that give the owner full control over the fruits of their ingenuity, or at least the right to prevent others from using it. Now, while the public are generally comfortable with the concept that the inventor of a new corkscrew, for example, should be able to patent and therefore restrict the use of their new invention, there is much less sympathy within the public with the idea of a scientist discovering a new gene in the human body, for example, and then filing a patent application to take ownership of that new gene. This is often thought of as taking ownership of parts of people's bodies and as immoral. Now, in reality, the public fear of someone owning part of their body is unfounded, as the requirement that a patented invention is novel prevents such claims. Now, you cannot patent something that is found in nature in the manner in which it is found in nature. Such a thing would not be new and would merely be a discovery, which, as you've learned from our earlier talk on patents in general, discoveries are not patentable as such. Therefore, as a bare minimum, any patent to a biological molecule or a biological process must be to a form of that product or process that is not naturally found in nature. Now, this ensures that the patent is novel, but this is only the beginning. With pharma, we're talking about small molecules to be used as active ingredients in medicines. And with biotech, we're more often talking about not only large biological molecules, such as antibodies and other proteins as active pharmaceutical ingredients, but we're also talking about medicines based on whole cells, such as CAR-T and stem cell therapy. We're also talking about vaccines, self-signaling molecules, gene therapy constructs, probiotics, and all sorts of other means for replacing, modulating, or removing aspects of the complex biological systems found in humans and animals. In addition to this, we have diagnostic methods, biological manufacturing methods, and medical devices, to name but a few. So let's look at the types of invention. So broadly speaking, what kinds of things can be patented in biotech and pharma? There are, generally speaking, 4 types of invention you typically see in the medicinal, biotech and pharma field. Now, these are chemical and biological materials, i.e. products, such as small molecule drugs like aspirin, large molecule drugs like growth hormone, and large biologics, such as Herceptin in this case, which is an antibody. Next, we have medical treatments and diagnostic methods. In other words, how you administer the drugs and to whom, and identifying who needs that treatment. And next, we have biologically implemented inventions, i.e. using biological systems to make things. Such industrial processes are crucial for manufacturing today's biological drugs. And last but not least, we have kits and devices used in the implementation of medical methods. Such medical devices usually perform a mechanical role in the body, such as keeping a blood vessel open. And here we have a picture of a stent. Special legal restrictions apply to nearly all of these different types of invention that are not generally found in other fields of endeavor. The exception, perhaps, are the kits and devices such as stents, which, being mechanical devices, generally have the same patent rules as any other mechanical invention. So for that reason, I won't discuss them in much detail here as time is short. With the first three types of invention, in addition to the need for novelty, European patent law has been drafted with special specific restrictions aimed at ensuring that patents are not awarded for inventions that would generally be deemed immoral by the public at large, or which would hinder the work of a physician in treating a patient in need. However, these statutory exceptions from patentability are quite narrow, and provided the patent is drafted with these in mind, it's usually possible to identify patentable subject matter where the inventor has genuinely discovered something new. Now, here is a point of divergence with practice in the US, where patent law doesn't explicitly restrict what can be patented, but where secondary legislation and a significant body of case law has been developed, which has similar aims, but often results in different outcomes. There are a lot of quirks in relation to how patents in these fields have to be drafted and the ways in which patent claims need to be structured, but I won't go into this in detail in this talk. Instead, I'll provide a brief overview of the main points. Now, European patent law states that patents cannot be granted to the human body at the various stages of its formation and development, and a simple discovery of one of its elements, including the sequence or partial sequence of a gene. You can, however, patent such elements if they are isolated from the human or animal body or otherwise produced by means of a technical process. Now, this includes the sequence or partial sequence of a gene, even if the structure of that gene is identical to that of the natural gene. Therefore, patents in its area have to be directed to the isolated molecule and preferably to a form that is modified in some way, such as by a mutation or by its presence in a DNA vector, such as a virus or a plasmid. The same applies to the newly discovered proteins, which must be isolated from their natural environment, or preferably recombinant, in order to be patentable. And patents for new monoclonal antibodies can be for the antibody CDR sequences, the full protein sequence, or even the hybridoma cell used to make the antibody. It's important to note, however, that unless you know or suspect how the new protein or gene will be useful in industry or medicine, then you do not yet have enough information to get a patent granted, probably. And when it comes to inventions involving microorganisms such as bacteria, there are very few statutory restrictions, other than the requirement for novelty, inventive step, and sufficiency of disclosure as per usual. It is possible to patent whole eukaryotic cells, but again, these must be isolated and typically be modified in some way, for example, to express a recombinant protein. An example of this is a CAR T cell, which is a T cell that's been modified to express an artificial T cell receptor. Now, when it comes to plants, genetically engineered plants can be patented, but not new varieties obtained through biological processes such as crossing. Instead, there are alternative IP rights, known as plant variety rights, that can be obtained to protect your new varieties of plants. Now, processes involving human and animal cells. This is, the European patent law sets out some very specific situations involving human cells where patents cannot be granted. Now, these are, as nicely demonstrated by the film The Island, processes for cloning human beings, no, not any animal, are not patentable. For those of you who haven't seen The Island, I recommend it, but some of the technology would fall within this exclusion. Another great film, Gattaca, processes for modifying the germline genetic identity of human beings are not patentable in Europe either. And Gattaca, another film certainly worth watching, the processes for making enhanced humans would not be patentable. Also, the uses of human embryos for industrial or commercial purposes are not patentable. And processes for modifying the genetic identity of animals, which are likely to cause them suffering without any substantial medical benefit to man or animal, and also animals resulting from such processes, are also not patentable. And these mice have been modified with a fluorescent protein obtained from jellyfish. Now, such mice might be patentable, but it would depend on the purpose of the experiment. It must be for a medical benefit rather than entertainment, for example. So there is a bit of a question mark when it comes to modified animals, whereas with modified humans, it's very clear this is not patentable. Now, even though you can potentially patent modified animals, there is a lot of ethical objection to this, and you would tend to find that your patents will be heavily opposed by various interest groups. But it's noteworthy that these exceptions all relate to processes and uses and not products, i.e. cells themselves. Nevertheless, it has been established by case law that totipotent human stem cells are not patentable, as they are considered part of the human body. Now, these are stem cells that can form any part of the human body. Now, further, the patenting of any other stem cell, where their provision necessarily involves destruction of a human embryo, is not permissible. Now, due to the availability nowadays of cell lines, it's generally possible to find suitable cells to work on without going back to a new human embryo for material for your experiments. So actually this objection in patent law is less common now than it was, for example, about 10 years ago. These prohibitions are aimed at deterring experiments that are deemed unethical, but of course there are multiple other forms of legislation that prohibit this type of experimentation. It should be borne in mind that a patent is a right to prevent someone from doing something, rather than a right to do something. So preventing patents is not necessarily a strong deterrent for this research. Indeed, it would seem that allowing patenting in this area might actually be more effective at restricting research. Nevertheless, this is an ethical debate for another day. Now, next we have medical treatments and diagnostic methods. It's not permissible in Europe to patent methods performed on the human or animal body. Now, this includes both therapeutic and diagnostic methods. But interestingly, non-therapeutic methods such as contraception or cosmetic methods, like injecting Botox, are actually okay to patent. And it is allowable to patent diagnostic methods that are performed on a patient's sample rather than being performed on the patient themselves. So providing your patent doesn't involve the step of taking the sample from the patient, then you're generally okay. The principal reason for these exclusions is to ensure that the patent system does not hinder the diagnostic or therapeutic activities of a doctor or nurse in a clinical setting. The European system does this by preventing patenting of the methods, but in the US, alternative mechanisms are in place to prevent doctors from being sued. So it is okay to patent methods of treatment in the US. Diagnostic methods are not patentable in the US at all. Instead, you have to direct your patent to the diagnosis plus the treatment step. So you have to essentially claim a treatment. The diagnosis on its own is considered to be a natural correlation, and that is not patentable anymore. This is actually relatively recent case law, as these methods used to be patentable in the US until the US Supreme Court decided otherwise. It is permissible to patent diagnostic methods in Europe, purely diagnostic methods, providing the patent does not claim the step of obtaining the patient sample. So patents to medicines in Europe must be directed to the actual product and not the administration of the product. Now, when the invention is the new use of a known medicine, this can clearly present a problem for obtaining a patent, as another patent to the same medicine would lack novelty. And as you can only patent the medicine, this could present a problem. So to compensate for this, it is allowable to patent new uses of known medicaments in Europe, provided the patent is directed to the product for that use and not the use itself. This is a subtle difference, but ensures that the patent is aimed at the activities of the drug manufacturer and not the doctor. Medical use patents can go further than just new indications. They can be directed to new dosage regimes or narrower subpopulations where the new data has shown that the drug will be more effective. Also, new administration routes and other features that can lead to a more effective medicine can provide you with new use patents. These types of claims are actually very commercially important in the current market in view of the push to repurpose abandoned drug programs. This can save applicant companies a lot of the costs and time involved in carrying out the early phases of drug research and drug development, and ultimately means that resources that went into such programs previously and were abandoned were not wasted. The next class of inventions are biologically implemented inventions. Now, biologically implemented inventions are typically methods for manufacturing useful products using biological systems, which is, they may be medicines, foods, or indeed industrial chemicals, for example. And for this reason, this area is often referred to as industrial biotechnology. Now here we have a system for manufacturing insulin, and we have some vats here where the bacteria making insulin are being grown. Now, there are generally no exclusions from patentability to worry about in this field unless the biological system being used is a eukaryotic cell, in which case the exclusions relating to parts of the human or animal body would apply, or potentially indeed the use of embryos, but this will of course depend on the exact nature of the cells you're using and how you obtain them. Now, if the invention is an engineered bacterial cell or a new cell-free eukaryotic expression system, for example, provided it is new and inventive, you are unlikely to have patenting issues. Here you could be patenting a new modified organism or culturing methods or media, for example, as well as the processes for making the desired products, of course. Now, there does seem like a lot of exclusions to patentability in Europe, but generally speaking, for any given innovation in the biological sciences, a discussion with your patent attorney will reveal that actually there are likely to be aspects of that are patentable, providing the system or the cells or the products you're using are actually new. So there's usually a way around these exclusions that will provide you with a patent that will help support investment in your company. Right, well, if you'd like to find out more or have any questions, then please get in touch, and the contact details are on this slide. In part 2 of this talk, I'll be talking about the data requirements before filing your patent application. Thank you very much.